See, this is what we are discussing about. I was talking about chromosomes and chromosomal aberrations. See, we began with connecting with the topic 1.7 biological basis of life, where we have studied about the cell and various components of this cell. So within the cell there is nucleus, and within the nucleus there is a material thread-like structures which we call it as chromatin. And when you take the condensed form of chromatin, you will get the chromosome. So chromosome is nothing but condensed form of chromatin. Now these chromosomes are nothing but this kind of twisted structures, and if you extract them, they will be like unwinding to give you DNA. So these DNA molecules are bound in the chromosomes with the enzymes histines. Now within this DNA, if you take a segment of it, a segment of base pairs which are orderly arranged, which are coded for that sequence, that particular sequence is called gene, and this gene is the unit of heredity. This is what I was talking about, and which we have covered it under 9.1, 9.2, even 9.3 as well, right? Now this DNA deoxyribonucleic acids, this is the composing material for your chromosomes, right? And a segment of DNA molecules, that is nothing but your gene. Like here it is coded also, like look at this, from here to here they are writing it as gene. That could be the code that has been a sequence that has been coded for this particular gene. And this entire information we have covered it in lower classes as well, earlier classes. So this is a chromosome. Now what is the structure of the chromosome? This also we have covered. It looks like a double what do you call this, dumbbell shaped almost, right? So it will be connected at central point. It will be having this kind of structure where each of these is called chromastid, sister chromatids. Each of these is called sister chromastid. Okay, and center we call it as centromere. Now depending upon the location of the centromere, we identify or we classify these chromosomes into 4 types. Can you tell me what are those 4 types earlier we have discussed?
Metacentric, telocentric,
जोर से.
metacentric, telocentric, acrocentric.
Yeah, metacentric, submetacentric, and acrocentric and telocentric. Out of these 4, which one is not found in the humans?
Acrocentric.
No, telocentric.
Telocentric.
Yeah, telocentric is the one which is not found in the humans, which we see it only in the animals, right? So metacentric, where your centromere is at the center and both the centrioles are almost at the equal. Now submetacentric means there is no slightly shifting of this centromere towards one end. As a result, you get one shorter arm, one bigger arm. Like this is called arm. We even call it as P arm and Q arm. P arm and Q arm. All right. And then third category is acrocentric, where centromere even further shifts towards little bit towards the edge, and this kind of acrocentric ones we find it in humans. Then there is a telocentric, which is completely at the extremity of the edge of one of the sister chromatids. That kind of structure we call it as telocentric. So 4 types. What are they? Metacentric, submetacentric, acrocentric, and finally telocentric. So these are the types. This also we know. This is very basic, which we have covered earlier. This is the reason why I was covering very elaborately there, so that we don't miss any of these. Okay. Now with this background, we can go ahead and look at the normal human karyotype. So karyotype is nothing but systematic arrangement of the chromosomes. So if we take the condensed chromatin and separate chromosomes and take the photograph of it or sketch of it and arrange them, that is called the karyotype. This is a karyotype which is showing of a male person. How do we know it is a male or female? This one. This is called sex chromosome. And remaining all 22. How many pairs of chromosomes are there in humans? 23 pairs, out of which 22 pairs are identical. Look at this. This one, this. See, each segment is looking same, right? So these are called autosomes. So we have 22 pairs of autosomes, and there is one chromosome which determines the gender of the person, which is called sex chromosome. Since in the sex chromosome we are getting the composition XY, so it is a male. Similarly, instead of it, if we get XX, then it would have been female. Now, why we are writing it as normal male? Because within these chromosomes, set of chromosomes, there are no errors. You can see it, there are no errors at all, right? That's the reason why normal male. Similarly, if it is XX without errors, we would say normal female. So this is what karyotype of human chromosomes. We are familiar with this as well, because in genetic studies we did study about karyotyping, right? Chromosomal and karyotyping methods. Now moving on, within these chromosomes there can be some aberrations. Aberrations meaning errors. There is a possibility of having abnormalities within them. Now, if you look at this, earlier we have seen the structure of the chromosome is something like this, right? And in case of numbers also, we have seen chromosomes are 23 pairs, and within this also you find like यहाँ पर भी डबल होना चाहिए, पेयर्स के जैसे होना चाहिए. So there is a possibility that in the structure as well as in the number there can be some errors, right? There is possibility, and these are classified into structure में अगर error है structural aberrations, वही numbers में error आता है numerical aberrations or numerical errors. Again, structural aberrations can be classified into 4 types. क्या क्या है? Deletion, where we will discuss about it clearly, where a portion of the chromosome or the complete chromosome got deleted. Duplication, where structure got duplicated, you get one more. Then inversion, where our arm has been inverted. Then translocation, where one chromosome moved from one location and gone to another location. This is called translocation. We will discuss about them very elaborately. Then comes your second category, numerical errors. Generally, you get chromosomes in pairs. That means 2 at a time. This is what we get. But suppose say instead of 2, you are getting only 1. Then there is a numerical error. This kind of numerical error, generally we call it as monosomy. Like pair hona chahiye, but instead of pair, there is only 1. That kind of error is called monosomy. Then second, let's say instead of pair, there are 3. मतलब एक ज़्यादा है. This condition is called trisomy. Generally, monosomy and trisomy occurs at one site, at one of the locations. Okay. And second thing is, instead of pairs at all the 23 locations, we have 23 pairs. Instead of it, if we have triplets at 23 locations, everywhere you have 3, 3, 3. Then it is going to be called, this condition is going to be called triploid. So you can have polyploidy, that is multiple haploids, like more than two. Diploid is the word generally we use it for normal chromosome which occurs in the pairs. So if something it goes beyond it, we call it as diploid. Or anything which goes below this 2 number, we call it as monosome, right? Complete deletion or something. So these abnormal numbers can also give you some errors. So that is what we will be looking at. Okay. Now moving on to see the structural errors. Like I told you, there are 4 kind of structural errors. Number 1, deletion. See, first of all, what are structural chromosomal aberrations or errors? How they result? They result due to breakage of chromosomes, breakage and abnormal reunion. So when does this happen? When there is cell division. We have studied earlier, under cell division what happens? There is non-disjunctional divisions. We have seen mitosis, meiosis. In both processes, there is a stage at which the chromosomes are going to divide into the daughter cells. If the division is not equal, then what will happen? There will be some numerical errors. While dividing, if there is any breakage, abnormal reunion of the chromosomes, that will result in the structural errors. Now look at this, deletion. See, deletion means delete karna. Like this, loss of some portion of the chromosome. Look at this. Here, this is not a healthy chromosome. Healthy chromosome should have been like this. Like it should have had both the portions. But from here, if let's say a portion of it got deleted, only like this, one of the arms got deleted or a portion of the arm got deleted. It's supposed to be like this, but this portion got deleted. Then we say there is a deletion, which you can see it in this particular one. So this is called deletion. It is a structural error. Then duplication. See, duplication means there is extra piece of chromosome, see, which you don't want it, but you get it a small extra piece. For example, this is a healthy chromosome. Let's say you are getting an healthy chromosome. On top of it, there is some more a portion, extra portion, which you did not need it. So this kind of extra piece of chromosome, or see, if there is extra segment of it, then it could have been numerical error. Like instead of this 2, if there is another one, another chromosome like this, it could have been a trisomy. But we are not discussing about it. We are discussing about the structural error where there is small extra piece which got attached to the chromosome, which is not intended. This also, when does we get it? When there is a breakage in the chromosome or abnormal reunion in the chromosome, right? This is called duplication. Then there is another structural error, जिसको हम कहते हैं inversion. What is inversion? See, fragmentation of chromosome followed by a reconstitution with a section inverted. देखो यहाँ पर मे भी थोड़ा सा समझने में मुश्किल हो सकता है, but look at this diagram, you will understand it. See, normally this supposed to be the normal chromosome and this is another chromosome. अब क्या हुआ है, यहाँ से this particular portion got like broke and this went and attached here on the other chromosome. So what happened here? This translocated from this location to this location, which is again not intended. So such type of error is called translocation. So third portion. Then finally, okay, we were discussing about translocation or inversion. We were okay, I was discussing about inversion, but I explained the translocation. It's okay. Anyway, we also need it. See, translocation is this, where a transfer of the chromosome, a segment of the chromosome happens to another chromosome. Here, a portion of it got broken and it has translocated to another chromosome. This is called translocation. Now what is inversion? See, it got broke here, and instead of moving to another chromosome, यहीं पर क्या हुआ था, उल्टा होकर. Like, let's say, is it visible? Is side if I draw it, or am I blocking it? Tell me.
Visible.
I'm not blocking it, right?
Yes, sir.
मतलब ब्लॉक कर रहा हूं कि नहीं कर रहा हूं?
No.
हाँ, ठीक है. सो अगर ये नॉर्मल क्रोमोसोम है, मान लो यहाँ पर ये वाला पोर्शन ऐसे जो है, आई एक्सप्लेन बोथ ऑफ देम हियर, ऐसे ये पोर्शन ब्रेक हुआ है. अब ये जो ब्रेक हुआ पोर्शन जो है, एक कंडीशन ऐसे हो सकता है कि यहां से ट्रांसलोकेट होकर दूसरा क्रोमोसोम के ऊपर लग सकता है, यहां पर लग गया. दिस इज कॉल्ड ट्रांसलोकेशन. मतलब अपना लोकेशन को एक जगह से दूसरा जगह कर दिया है. अब दूसरा कंडीशन ये है कि यही वाला क्रोमोसोम ब्रेक हुआ है, बट दूसरा क्रोमोसोम पे नहीं गया है. यहीं पर अर्लियर ऐसे था, अब ऐसे उल्टा होकर उसी पे अटैच हो गया. ठीक है, पलट के. लुक एट दिस हियर, this segment of it is clearly shown like it got broken. यहाँ पर ये वाला पोर्शन जो है ब्रेक हुआ है, and it got attached here, this extension. ठीक है, this kind of structural error is called inversion, right? So once again let me summarize it. बीच में कुछ confusion आया था ना, थोड़ा इधर उधर चला गया था. Deletion का मतलब completely removal of the material, DNA material from the chromosome. Sorry, it can be a portion of the chromosome or complete segment of the chromosome, complete arm of the chromosome, right? Depending upon how much portion of the deletion is there, उतना खतरनाक diseases आएंगे. Then comes duplication. Duplication का मतलब है कि इसी टाइम पे when there is division of these chromosomes, there is like abnormal reunion which is giving some extra material, extra chromosomal material on one of the chromosomes which is not required. So this is duplication, extra piece of chromosome. Then third is inversion, where within the chromosome a portion of it got separated and inverted, reattached to the same chromosome. It did not go anywhere. मतलब ब्रेक हुआ है, सेपरेट हुआ है, फिर उल्टा हो गया है और उसी क्रोमोसोम पे लग गया है. दिस इज कॉल्ड इन्वर्शन. देन ट्रांसलोकेशन, अगेन ब्रेक हुआ है पोर्शन और कंप्लीट आर्म ऑफ इट, बट यहां पर उसी क्रोमोसोम पे नहीं है, कोई और क्रोमोसोम पे कुछ और लोकेशन पे जाकर अटैच हुआ है, वो भी विदाउट इन्वर्टिंग. देन वी कॉल इट एज ट्रांसलोकेशन. These are the 4 types of structural errors in the chromosomes. Okay, then numerical errors. What are numerical errors? Numbers में error आना चाहिए. Ideally, as per the ideal composition of the chromosomes, we must be having 23 pairs of chromosomes. 23 pairs मतलब 46 chromosomes होना चाहिए. This is the ideal case. Now any deviation from this 46 number, let's say 45 है, मतलब एक जगह पे 1 chromosome is not present, और 47 है, एक जगह पे एक क्रोमोसोम एक्स्ट्रा आ गया है, या 69 है, मतलब हर जगह पे let's say 23 pairs के जगह 23 triploids like triplets है. ठीक है, then you will get 69. This is also deviation from this ideal constitution of 46. So any of such abnormal condition can be called as numerical error in the chromosomes. And why does this happen? Again, the reason is same. In the cell division process, in the meiosis process, mitosis process, when there is like a division of the chromosomes, if the divisions are not perfect, like you can say unsymmetric disjunctions are there, that is when you will be getting some chromosomes extra in some of the cells, and that will lead to this kind of problems. ठीक है, I'll explain it with the diagram now. ठीक है, now look at this. They are type of chromosomal defects and generally birth defects है, and where the composition differs in the number of chromosomes. So instead of typically 46, we will be having either 45, 47 or sometimes 48 also. ठीक है, in super females also you get multiple X syndrome. ठीक है, we will look at them. ठीक है, what is the differences and all the characteristics and all. So mechanism is simple. This is the normal cell division process. Look at this. From the parent cell, the daughter cells are perfectly getting that material and disjunctions are happening. And even the next stage, phase 2 also is getting equal number of chromosomes. But in the abnormal conditions, it can happen. Look at this. Here itself, you have disjunctions. So this is one type. Another type is in the meiosis 1 process, there is good division, but in the second process, there is abnormality. Look at this. Here, you are getting the abnormalities. Here, all of them are abnormal. Here, 2 of them are okay, but remaining 2 are having abnormalities. This kind of abnormal division of the chromosome is the reason behind numerical errors. Now, these numerical errors can happen in the autosomal chromosomes, also sex chromosomes. Please remember one thing I forgot to mention. Generally, structural errors only happen in the autosomal chromosomes. Sex chromosomes में नहीं दिखता है ये structural errors. Sex chromosome में क्या prominent है? Numerical errors. So numerical errors can be seen in both autosomes as well as sex chromosomes. दोनों में मिलेगा. ठीक है, दोनों में ये एरर्स मिलता है. तो यहां पर देखो एग्जांपल्स भी दिया है. ऑटोसोम्स में यू मे हैव ट्राइसोमी at 13, ट्राइसोमी at 18th लोकेशन, 21st लोकेशन. उसी के हिसाब से अगर 21st पे है यू गेट डाउन सिंड्रोम. वी विल डिस्कस दोस ऑटोसोमल एरर्स डिसीजेस एज वेल टुडे. ठीक है, सिमिलरली दिस कैन हैपन monosomy also. One of the chromosome can be missing. For example, Cry-Du-Chart disease. P location जो है 5p माइनस बोलते हैं उसको. तो 5th क्रोमोसोम पे PR मिसिंग होता है उसमें. ठीक है, that is the reason why you get this disease. Similarly, you can have the structural errors in the sex chromosomes. Ideally हमारे पास इधर सेक्स कंपोजिशन में XX होना चाहिए या XY होना चाहिए. दिस इज द आइडियल कंपोजिशन. समटाइम्स X के जगह मल्टीपल एक्सेस है. ठीक है, और X 0 है, मतलब दूसरा वाला X जो है दैट गॉट डिलीटेड. ठीक है, दैट मीन्स यहाँ पर 46 के जगह 45 ही क्रोमोसोम्स है, क्योंकि दूसरा वाला X जो है दैट इज डिलीटेड. ठीक है, दैट इज नॉट एट ऑल प्रेजेंट. Then also you call it as like monosomy. This will give you Turner syndrome. Similarly, Klinefelter syndrome, जहां पर XY is the composition in the males, but instead of it, multiple X and Y comes, right? This only comes in males. Please remember, Klinefelter syndrome जो है ये only males में होता है. Similarly, Turner syndrome जो है वो only females में होता है, क्योंकि composition इस तरीके से है. So these are all some of the examples of numerical errors, right? So let's summarize what we have discussed so far. Chromosomes, what are chromosomes? The location of them, the structure of them, and the types of them based on the location of centromere, the 4 types that we have seen, and structure we have seen. Then we have gone to the abnormalities or errors in the chromosomes. And we have seen what is the cause behind it. There are multiple causes, but main one is cell division. So if there is any imbalances while having the division of cellular material, particularly nucleus material, that is when you get this either structural or numerical errors. Apart from that, some mutations can also lead to this. Now, within these errors, you have two types of them, structural errors and numerical errors. Structural errors again classified into four types, where you get one is deletion, another one is translocation, inversion and duplication, where structure में कुछ ना कुछ मतलब abnormality दिख जाता है. Now next type is numerical errors. न्यूमेरिकल एरर्स मतलब नंबर्स में एरर होना चाहिए, और ये नंबर्स में एरर जो है अगेन दो टाइप का है. ये क्या है कि नंबर्स कम भी हो सकता है या बढ़ भी सकता है, एंड इट कैन हैपन इन बोथ ऑटोसोम्स एज वेल एज सेक्स क्रोमोसोम्स, जहां पर 46 आइडियल नंबर से डिविएट हो जाता है. इट कैन बी 45 और 47 और इवन मोर देन 47 आल्सो. You can have triploid condition as well. So this is another. And structural errors जो है वो मोस्टली हम ऑटोसोम्स में ही देखते हैं, सेक्स क्रोमोसोम्स में नहीं देखते हैं. और न्यूमेरिकल एरर्स जो है वो ऑटोसोम्स में भी सेक्स क्रोमोसोम्स में भी दिखता है. So these are all the points that we have discussed so far. Now tell me, is it clear everyone?